clear on that. So the field guide is due this Friday, right? And I think what I might have mentioned this to you, I'll probably create a drive folder for you, and then I'll share that to you so that you can upload into that, because I'm worried that the file size that you have for your field guide might exceed what EduCat allows. They only allow so many megabytes, and pictures take up a fair bit, especially if you don't compress them, which is a pain. So I think it'd be better to do the Google Drive folder. So I'll create that and send you a link. And then the Human as Habitats lab report, just of the results and discussion portion. I have that down that it's due next week on the 28th. Okay, the 28th. And again, I think a regular EduCat will be fine for that one. And then, let's see, the lab exam is due November, or will be November 4th, which will be during week 10. So this is week 8. So 2 weeks out, we have the lab exam in lab. And so I will send you information on that. So I did mention in lab the other week that I'll be breaking up the material that we've been learning weeks 1 through 8 so that it doesn't all show up on lab exam 1. So I'm going to be splitting that out. So between lab exam 1 and lab exam 2, the intention there is to cover material weeks 1 through 8. And then your research project is something very different. You will be like quizzed on that. That's a different thing. And then the lecture exam is on the syllabus for week 11 during the week of November 7th. I was thinking maybe Wednesday, because I think we have freedom either before or after class. No one's in here. So that way I could extend how long the time, so you have a little bit more time. Questions on any of those deadlines? Yes. New material only. New material only. And then we do have to start thinking about the research project. So some of you have talked to me about your general idea. So this Friday in lab, we will take a shot at analyzing the Winogradsky columns that have been percolating in the windowsill. And we'll see there's been some interesting developments just visually. So it'll be fun to look at them under the microscope on Friday. And then also during Friday, I will be circulating around and asking you about your research project. And so by Friday, if you could have an idea or a couple ideas that you would be interested in pursuing. And you don't have to come up with something super novel. You're not expected to be Einstein. And again, we have very limited budget. So I've had students come up with awesome projects, and unfortunately I have to say, we can't afford that because it would require special equipment or supplies. So think about things that you've been learning in lab already, and those types of skills you could easily incorporate into a research project. I also have additional, like there's other physiological tests, for example, that we didn't learn in this class. But if you've taken Gen Micro, as some of you have, you're aware that there are a bunch of other physiological tests that are optional. So as you talk to me about your project, I may suggest, oh, well, we could try this one physiological test, and it's something that is straightforward and easy to do. And so just because you haven't learned it already doesn't mean you can't incorporate it. We just have to be careful on costs, because usually I don't have as many students in the class, and so the costs kind of go up obviously exponentially. Excuse me. So if you could think about that between now and Friday, and I'll be chatting with you all on Friday. If you want to send something to me via email before that, that's fine too. All right. Yes. No, I don't do that anymore because then people will wait till 11:58 to submit, and I don't like that. I don't like you all working that late at night. And you don't do your best effort that late. So anyone, I'm not pointing fingers. I'm just saying the human body in general doesn't do well with that. So I think I usually make it. Let's see. I usually do 7 p.m. on Fridays. That way you can submit and forget about it and have a little bit of time on Friday to relax. All right. Is there well, can you get it done before you go to work? Okay. Yeah. I do have, but that reminds me, I do have, I accept late assignments. I do that. I have to take off a little bit of points, but I know some professors are very like, no, once the deadline. I can push it to 8. That's fine. And then just be aware if something comes up and you're delayed, don't not submit. Go ahead and submit it, even though it's after the deadline. It will accept. I set it up so it accepts late assignments. But try to make it. Any other questions regarding the field project or field guide? Yeah. Resources. So basically anything like when you're talking about certain groups of microbes, you need to cite where you got that information from. Like, how do you know that the water has phototrophic bacteria? What did you read? And so I need like a reference list at the end, and it has to be something I can readily access. Yes, the stuff in Educat, readings and videos. Yeah, that's why I posted those. Those are good enough. You don't have to find some super, yeah, yeah, that's fine. That's why I posted those resources for that kind of stuff. I usually say APA. I don't really care too much. The big thing there is to make sure each reference is in the same style. And so don't do APA and then what's the other one? Yeah, MLA. Don't mix them. Just stick with one. Is there like no, it's more the diversity of the site. So it's not just like all like native grass, just boom, that's all it is, and so if it has some diversity to it. In general, it's something that you can easily see within, I don't know, 50 feet or so. People usually pick something like that to look at and keep it around there. Some go a little bit bigger. Basically, you have to imagine me going to your site and walking around and looking as I'm reading through your field guide. And so I don't want to be tromping for miles. Don't send me on miles of a trek on your site. So it's usually I go and then I stand and I look around. I might walk around a little bit, but yes. Yes. I'm with you on that. I'm not artistic or creative too much when it comes to that. Some people are, and if that is something that you can incorporate, awesome, go for it. If that's not your forte, a simple Word document, that single word, that's fine, and that's what most people submit. So don't worry about that. And again, it's more the content. But again, if you're creative and have that capability, go for it. Yes. Yes. Yeah, so you'd say like figure 1, and maybe you took a picture of a mixed deciduous forest, and then you could just say, on this site you will find a mixed deciduous forest. Yep, yep. You just want to kind of summarize the image for the reader, and you don't have to get super detailed. But yeah, all figures should have, and you label them at the bottom of the figure, not at the top, figure 1, and then do a couple descriptive sentences, figure 2, and so on. Oh. So are you saying like if you're on your computer and you are typing it up as a Word document and then you upload it into Google Drive, it messes with it? Yeah, take it. Okay. Yeah. I know. Yeah, go ahead. Go ahead and take. Pardon? No, I mean, you include the best pictures you think are most relevant. You probably took more than what you'll include, I'm guessing. And so just take the ones that are most relevant to the field guide, the most exciting. Yes. Yeah. Go ahead and take the pictures. Mhm. It is what it is. I mean, we have to deal with Mother Nature. The other thing about inserting pictures that I've found in Word is I'll do a copy paste, and then in the, how does that work? And then it'll give you the option to, I think I right click on it and it gives all kinds of different options of how you want to insert or paste in this. And I always tell it to go over the words or something like that, and that fits it in really well. I can show you maybe after class if you're curious. I can't remember exactly the steps, but I kind of, once I start doing it, I just have muscle memory and I can show you. Okay, so we are looking at biogeochemistry, which was last Friday. I think we left off here. So I do want you to study this and be comfortable with this material presented in this format. I think it's a nice visual to really succinctly summarize what can happen both in the presence and then in the absence of oxygen. And so we went through this on Friday where the presence of oxygen is up top above the black horizontal line, and then anaerobic is below it. And so looking at one of the ideas on here, I wanted to go into a little bit more detail where you see down here the carbohydrate at the very bottom of the image. And they're saying that you could have methanogens running it that way, but you also see CO2. There's a line coming off CO2 and running to methane. And so I wanted to talk a little bit about what's happening on the left part of this circle where you have the methanogens acting in an anaerobic environment, and then the methanotrophs acting in an aerobic environment. And so gen means genesis. And so we're looking at the creation of methane. And so far, only cells or microbes that are in the domain archaea seem to be able to do this. And they are poisoned by the presence of oxygen. And so they're strict anaerobes. And so they're very good at taking CO2 and reacting that with hydrogen gas, which is common in anaerobic environments, both of those. And when you do that, HCO3 minus is just how we denote CO2 when it's in an aquatic setting. When it dissolves in water, it's HCO3 minus. And then you can see the reaction here. You don't have to memorize the exact reaction. Just know that they can take CO2 and react it with H2 to form CH4, methane. And why are they doing this? Because it provides them some good free energy. And that free energy can be used to do something. Often it's done to make ATP. So it's exergonic. That means if you have hydrogen gas and CO2, it's going to react spontaneously and give you some nice free energy. And so they're running it because they can get ATP off of that free energy. It allows for them to develop a proton gradient. So they use an electron transport chain, ETC, electron transport chain process, and they will link that to pumping protons across the membrane, which will lead to a proton gradient and ATP production. Proton motive force is a fancy way to say proton gradient. I like proton gradient. It's a little bit more precise of what we're talking about. They can grab acetate as well. So that's why on that figure you saw that the arrow was coming off of the carbohydrate to methane. So an acetate molecule would be an example, or methanol, or methylamines. So they can grab some simple other substrates and run a reaction to get methane. Yes, so they are incorporating both hydrogen gas and the proton. Yeah, they can do that. Let me show you. I think I have a picture that might help. So they're doing all of this anaerobically, but then it's usually safe to say that if you have methanogens present, there'll be methanotrophs lurking nearby. And that's because they kind of do the opposite, where they'll take the methane. So remember, when methanogens are making methane, their goal is not to make methane. They don't do anything with that methane. They just excrete it. They're just running this reaction to play the game of electron transport process so they can get a proton gradient. That's their goal. And they want to do it in such a way that it allows for some good ATP production. So their goal isn't methane. They just excrete that. So the methanotrophs lurking nearby in an aerobic area will go, well, I can use that. I'll take the methane and strip electrons off of that. So they take it and use it as an electron donor. And the carbon can also be used as a carbon source that they can build carbohydrate off of. So again, you don't have to memorize in totality the reaction, more just what you start with, what you end with. All right, so here you see methane being in a series of steps being broken down. So electrons are getting stripped and you end up with CO2. So essentially it's an oxidation. You're stripping electrons in a sequential fashion. So let me show you a picture. And again, you don't have to memorize step by step. It's the big picture. So here is methane on the lower left and the sequential steps here. And so they're linking that. They're trying to show that it's linked here to an electron transport chain process. And that allows for a proton gradient. And so you can see that often there's free protons involved in getting this all to completion. So having both present, it's just that one is an obligate anaerobe or strict anaerobe, and then the other one is in an oxic environment. And so this diagram is trying to get at that idea where you tend to see this type of interaction in lakes, where in the sediment, the methanogens will be in the sediment, and because that's tends to be very anaerobic. And they're making methane, it's a gas, so that just kind of diffuses up into the water column. And then you have the methanotrophs capable of using the methane. Here you see, well, anaerobic, there are a couple of species of anaerobic methanotrophs, but the lion's share of methane use is aerobic from what they're finding in reality. So here, this is an aquatic environment. I just put this in here because I thought it was a beautiful picture. This is a lake in Switzerland. Those are ice bubbles, essentially. Bubbles that were captured when the water was freezing. And it says in the notes section, these bubbles were likely formed from methane. Decaying organic matter at the bottom of the lake is eaten by bacteria, which in turn pump out methane gas. If conditions are right, the methane bubbles are captured as the water freezes around them. When the water ices over and the surrounding mountains are blanketed with snow, the name of this lake is particularly fitting. Lago Bianco translates to white lake. I just thought that was a real pretty visual, thinking about methane gas. Isn't that crazy? So lots and lots of methane, apparently, when you zoom out. Look at all that. So they're very, very busy, very happy down there, even though it's freezing conditions. They're still, they're psychrophiles. They've adapted. Okay. Another group here that the reason I'm talking about this group is that they also grab CO2. So I said that methanogens can grab CO2 and react it with hydrogen gas to make methane. So now acetogens are also doing the same thing. So if they're in the same environment, you'd think there'd be some definite competition going on. Domain bacteria, most of them are gram-positive that can do this. And so they're also, like the methanogens, they're also capable of grabbing CO2 and reacting it with hydrogen. And so they're both vying for the same substrates for their free energy needs. Acetogens, though, they're also a little bit more diverse in what substrates they can capture. Methanogens are more strict. They have very few options. Acetogens, on the other hand, they can use some sugars, AA or amino acids, alcohols. And so they're a little bit more, they have more of a stock in their pantry. There's more things to pick from if they're available in the environment. But they also, usually hydrogen gas is pretty abundant, and hydrogen is just really, really good at giving up electrons. And so reacting these two together gives you a nice free energy. Again, negative 107, that's pretty decent. And so that will definitely get you some ATP. ATP production, they generally say you want to get more negative than negative 32 kilojoules. So anything more negative than negative 32, and you're going to get ATP off of that. So negative 107 is definitely more than negative 32. More negative. So you have this situation where they're both vying for the same substrates. And when you look at it, the free energy is actually better if you get the methanogen reaction going, you get more free energy. So you would think if they're both in the same area and the methanogen are grabbing hydrogen gas and CO2, they get more bangs for the buck. They get more free energy, which means that their population levels will probably start to build faster than the acetogens. And so if you think about it, you could imagine a situation where if they keep running this more favorable reaction, they could maybe totally outcompete and replace the acetogens in that habitat. And when they do these studies, they find that that rarely happens because the acetogens are just a little bit better at being more diverse in their substrate options. Whoops. And so you have the acetogens, they're like, well, okay, you're hogging all the hydrogen, you darn methanogen, I'm going to take some amino acids or some alcohols and run it instead. And so when they run these reactions in the lab, and then when they go out into the field sites and look at what's happening in natural settings, they do see that they can coexist if there's enough of the substrates available for the acetogens. Even though they get less free energy, they don't get outcompeted typically. The other thing, acetogens, they're making acetate, which is an acid, an organic acid. So acetate, another word for acetate is acetic acid. So they create lower pH levels and they can tolerate that better than the methanogens. So that's another plus in their favor, even though they don't get as much free energy. So again, they tend to coexist, even though on paper it seems that methanogens should win and outcompete. Yes. The acetogens, is that what you're asking? Methanogens, I don't know if they've found any other than just strict anaerobes. Acetogens, I believe, are also strict anaerobes, but let me double check. They might be facultative. I'd have to double check that. Yeah, let me double check that. I might be remembering that. Carbon monoxide, thinking about the carbon and oxygen cycle again. I'm thinking about carbon monoxide and how that gets transformed and recycled. In the oceans, that's where you find most carbon monoxide being produced, and it's through algae and cyanobacteria that we see that. In the soil, it's actually a sink for carbon monoxide, so it gets fixed into carbohydrates in the soil by the actions of microbes. And so it's a carbon and energy source microbes can take advantage of. Even though it can kill us, they can use it. And they do that aerobically. So what that means is they're using oxygen as a terminal electron acceptor in this case. If they do it anaerobically, they'll usually react it with hydrogen gas and you get your methane. So methanogens are doing that. So carbon monoxide, even though it's deadly to us, it's actually something that the microbes are heavily involved in using and recycling it through a variety of means. So methane and then acetate. All right, so carbon monoxide kills us. Microbes are happy to use it. Polysaccharides, very complex molecules. Poly means many, so you think about like cellulose, hemicellulose, chitin, those are all extremely complex polysaccharides. And if it weren't for microbes, our forest would be building up in a bunch of dead trees and plants in general. So the big picture here is that microbes rule when it comes to breaking down these complex polysaccharides. Fungi are awesome at doing this. And these are just listing out some that have been heavily studied. You may be familiar with some of these genera if you enjoy reading about fungi. Bacteria are also really good at this. The Streptomyces bacteria that I work with in my research are famous for this. In fact, when I was a grad student and I went to present at a conference, there was a professor out at the University of Idaho in Pullman, no, right across from Pullman, Washington. Moscow, Idaho. That's where it was. And he saw my poster. I was working with streptomyces, and he wanted to have me as a postdoc in his lab because I had all that experience with streptomyces, and he was doing this kind of idea of how the streptomyces are important for carbon breakdown and cycling. But I said thanks, but no thanks, because I wanted to work with the idea of biocontrol. I was very interested in how the streptomyces could be used for biocontrol against plant pathogens. But he's published numerous articles. He helped write a book on streptomyces, so he's done a lot of research with their abilities. You can do both aerobic degradation of these complex polysaccharides, and again, fungi are really good at that, but so are many bacteria. And then anaerobic, it's Clostridium has been well studied for this ability to degrade polysaccharides. So unfortunately, you hear a lot about like Clostridium botulinum, Clostridium tetani, that can cause serious disease in humans, but Clostridium is in the soil. Farmers, for example, can get exposed to the Clostridium botulinum, but it normally doesn't kill you if you get it on the skin. It's more if it gets into your interior cavity that's the problem. But if it's on your skin, it's not usually a problem. Anyways, there's Clostridium in the soil, and they're very good at breaking down complex polysaccharides. Hemicellulose is a little bit different than cellulose. It's a little bit more complex. It's branched. And so, again, it's very challenging to break down, but bacteria and fungi have the necessary enzymes. And so, do I have, let me just check. Yeah. So when you think about plants, the plants have cell walls, and their cell walls are very full of cellulose. That's the main ingredient, cellulose and hemicellulose. And then some cell walls, we call them secondary cell walls of plants, they have lignin in addition. So there's a lot of complex polysaccharides out in the forest system that has to get broken down. Chitin, if you think about all the insects out there and their exoskeletons made of chitin, so part of the chitin molecule is N-acetylglucosamine, which is actually a component of the cell wall of bacteria as well. So the peptidoglycan that we talked about with the gram stain idea, part of peptidoglycan has N-acetylglucosamine. So this molecule is super, super abundant in our natural world. When you add up all the microbes out there and the bacteria and then the insects and fungi. And so we have a lot of that to be broken down. And again, the fungi, bacteria, and actinomycetes are very good at it. At least, well, I don't know if they've been able to pinpoint one as superior to the other. It seems like they're all pretty equally active in chitin degradation, which is a good thing because there's so much of it. And then lignin is part, again, of the secondary cell wall of plants. And so there's plenty of lignin out in our forests, and it's incredibly complex. It's more complex than cellulose and hemicellulose. It's a very branched polymer. They actually didn't know the exact chemical structure of it until fairly recently, because it's been very difficult to study and break down and figure it out in the lab. And so CW is cell wall, part of the secondary cell wall of plants. It's complicated and difficult. And so what they see is that if you have straight intact lignin, it doesn't seem to be able to be broken down in an anaerobic environment. There's been no microbes that seem to be able to do that. And so it's the aerobic bacteria, and it tends to take groups of different microbes in order to fully accomplish it. And so, there, for example, there's these groups of fungi called brown rot fungi. And what they're doing is they attack the cellulose and hemicellulose of the tree trunk. And then the white rot fungi can come in and start attacking lignin. And so it seems to only be done aerobically. We don't see the breakdown in an anaerobic state. And so those images are the brown rot and white rot tree trunks that you're seeing. And so breaking down cellulose, hemicellulose, lignin, and acetylglucosamine, it's usually many microbes acting in consortium as a team to attack this and get it fully recycled. So a lot of activity. Mm-hmm. Yeah, they can, not all microbes, not all bacteria, but many can. Mm-hmm. Fungi are well known, they're very good at it, but there are bacteria that can do this too, yes. So like the streptomyces that I work with, some of them are very, very good at that. It's just that fungi have kind of gotten the gold star for their abilities. There's been a lot of attention with fungi. And if I'm remembering correctly, I think they've discovered at least one fungal species that can break down plastic. Has anyone heard about that? Can degrade plastic. And so they're seriously looking at that, obviously, because we have so much plastic in our environment. So they're working on that, trying to see what they can do. So lignin, just trying to give you a visual to impress on you that it's very complicated, and it's without the microbes there, we'd be stuck. It's very challenging to break down. If you look in, so most of what I was just talking about are things that you think about in forested ecosystems where there's a lot of trees and tree trunks. And then thinking about aquatic systems, how would they break down polysaccharides in aquatic systems? It's usually not as abundant, obviously, because we don't have trees growing in the oceans. But so in general, a good statement is that you have much lower amounts of polysaccharides in aquatic systems. And but having said that, bacteria can work with that, and assimilate means that they can take polysaccharides, partially break them down, bring them into their cells, and use them either for ATP energy needs or to build biomass. So when they say assimilate, they're referring to building biomass when you hear that word. So they can take in, they'll break down the polysaccharide by excreting exoenzymes and then bring it in. And even though there's low amounts in the ocean, they're just really good. They're very efficient at being able to take in those carbon sources once they're broken down small enough to bring into their cells. And then they can use it either for ATP running, ATP production, or they can assimilate it into biomass, make more cell material, and it'll help them reproduce. So when we talk about carbon recycling, it's usually carbon, hydrogen, and oxygen that we kind of link all in one big idea. It's hard to separate those with how life exists here on Earth. And so, we have oxygen, CO2, water. When we think about, where is oxygen showing up in abundance? And then don't forget, like nitrates, and nitrogen-containing compounds have a lot of oxygen involved in there too. And when you think about the oxygen cycle, obviously a big part of that will be the production of oxygen by photosynthetic organisms like the cyanobacteria, the algae, and then the plants, that both, terrestrial plants. And then in the oceans, it's more the algae and the cyanobacteria. So oxygen is a big deal for life on Earth here. And as you well know, if we didn't have oxygen, we wouldn't be sitting here. And so it's a big deal for microbes as well. And I have been trying to make a point of this through the semester ever since we started talking about the physiology aspect of microbes. Whether or not there's oxygen presence can make a big difference to microbes in terms of the payoff. And so if the oxygen is available, the smart thing for a microbe to do would be to use an electron transport chain with oxygen sitting at the end and having that thing running so you can get some ATP production. But as you know, microbes can exist in anaerobic environments. Well, then the next best option in that case would be anaerobic respiration. Still use an electron transport chain because you'll get the best ATP production doing anaerobic compared to fermentation. And so if the microbes are in a situation where there's no oxygen, they tend to pick these types of molecules to put at the end of their electron transport chain. So we did mention nitrate quite a bit, but there are some other options, and they're all decent at allowing for a proton gradient to develop. And so it's the same basic idea. You have this electron transport chain, and the whole goal there is to create a situation where you can get proton pumping and get a proton gradient established. So then if you're a microbe and you're in, first it's like your facultative, oxygen's available, go ahead and pick oxygen. If that becomes unavailable, switch to anaerobic. If that becomes not a viable option because you've run out of a terminal electron acceptor, then you might have to switch to fermentation, which is not so good because it's not an electron transport chain. Or if you're methanogen, you could do methanogenesis, or if you're acetogen, you could do that too, do acetogenesis. So there's options out there. I would definitely want to be a facultative anaerobe if I was one, because that gives you some good flexibility. So we've looked at this before with the idea of aerobic respiration on the left, or aerobic metabolism versus anaerobic metabolism on the right. Just jogging your memory on that concept. And then fermentation, we've looked at something like this already, where it's not electron transport, it's substrates that are transferring the phosphate instead and getting phosphate transferred to ADP to make your ATP. So much lower ATP production, you don't get as much free energy out of that. So this is kind of putting it together with the carbon, oxygen, and hydrogen cycles, and looking at that and just thinking about how really when you get into any of these cycles, whether it's carbon or nitrogen, and in lab we'll talk a little bit about the sulfur cycle when we look at the Winogradsky columns. Microbes are just dominating this whole idea of getting these different compounds recycling on Earth. The nitrogen cycle I did want to mention in addition to the carbon cycle because it's pretty important, and especially when you think about all the microbes, it's super critical. They play such an important role. So nitrogen is, you can strip electrons off of nitrogen pretty well. It gets you some nice free energy. And really, if microbes didn't exist on this planet, we wouldn't have a nitrogen cycle. We're messing with it pretty considerably because of what we're doing with our industrial processes. So you may have seen something like this in an intro ecology class, and I'm not really expecting you to memorize all of this. A couple of key points that we've been kind of thinking about is that you have the nitrogen gas in the atmosphere, and prokaryotes are the only ones that can grab that. And so again, if we didn't have microbes, the prokaryotes, this Earth just wouldn't be the way it is. And so you're seeing the nitrogen up here getting fixed to the biological nitrogen fixation. And then, but you also see that once they have nitrogen forms in the soil, there's a couple of different processes going on here too, where you end up actually getting the reverse happening, where you have the nitrogen being denitrified and becoming N2 again. All right, so we've talked about this a little bit already when we looked at this slide in our physiology section. So the denitrification idea is if you have microbes that are, let's say they're in the soil and they have nitrate, they'll run this reaction. So it's an electron transport chain. They're doing it to get a proton gradient. And then some microbes can only take it so far. Others, like Pseudomonas, can take it all the way to dinitrogen. So I wouldn't want this in my garden because it's doing the exact opposite of what I would want. I want my plants to have nitrate, not for nitrate to disappear. So definitely wouldn't want this in a garden or agricultural soil. And so, you know, the vast majority of our nitrogen is up in the atmosphere. 80% of our atmosphere is nitrogen by nitrogen. So only prokaryotes can do that. The only way we can do it is a very powerful synthetic process called the Haber process. Do you learn about that in chemistry, maybe? I don't know if that's something they teach. I think, isn't it the Haber-Bosch process or something like that? It's named after the scientist that figured it out. So this one I'd like you to spend some time with, all right? More so than the other picture that was really complicated. So I was showing you this one, but I prefer that you zero in and think about this one, all right? So this is famous. They like to present it this way as the triangle, where we just talked about how microbes in the soil, we don't want them in your garden to take nitrate and convert it to dinitrogen. That's denitrification. I would like you to know the terms, please. And then on the other hand, if you grab dinitrogen and create ammonia from that, that's a fixation, nitrogen fixation idea. And then if you take ammonia or ammonium and run a reaction that gets you nitrate or nitrite, that's the nitrification process. And so microbes are doing this, you know, different groups of microbes are doing this and keeping this all going on Earth. And so again, if it weren't for the microbes, it just wouldn't happen. Here they're just showing it again in a slightly different form. A little bit busier figure here, but it's good for studying if you are trying to make sense of this. Maybe this slide will help you work through it. Okay. So I could go on and on and on with biogeochemistry because microbes rule. They just dominate that whole idea. But we're going to stop there with this topic. And then on tomorrow in lecture, we'll get into species interactions, think a little bit about that. And then after that topic, we'll get into some specific microbe or other organism interactions. All right, we'll stop there.